So I'm in chemical engineering and material science, as was mentioned, but I'm also in electrical engineering and biomedical engineering and the Keck School of Medicine. So my group does a lot of different things, but my group has 14 grad students, 7 undergrads, and 3 postdocs at this point. So it's very large, which helps do all of this stuff. Working on coming up with new types of lasers, integrated devices, which I'll talk about in a minute. We have another section of the group working on coming up with new types of materials, so inventing new types of glasses, new types of polymers, coming up with ways to put the materials onto a silicon wafer, which then feeds back into the device work, because if you can come up with a new material, you can then make a new device out of it. And then a third portion of the group works on studying how molecules behave, how proteins behave, how cells interact with each other as you change the environment around them. Conventionally, how people do these studies is they use a microscope, so a fluorescence microscope is kind of the standard way of doing it. However, fluorescence microscopes have a lot of limitations. Namely, you have to put a fluorescence probe on whatever protein you're trying to study. Fluorescent probe is a little tiny molecule that emits light. So what we're trying to do is use our devices to make these measurements a lot easier, faster, and make it so you don't have to have that fluorescent molecule on there, so you can do it without a label or without the fluorescent probe. So basically everything kind of feeds back on each other. So we come up with new devices, make new materials to make the devices better, and then use the devices to study how molecules and cells behave. So some of the optical device work. So the conventional integrated electronic circuit looks something like this, if you're looking at a picture from 1995. And what we're working on doing is creating an optoelectronic circuit. So basically you have an optical layer, which then interfaces with an electronic layer and a processor layer. So you would need something on here that would emit and receive light, because you have a photonic layer on there. So there's a Moore's law shrinking that says that every couple years the size of wires on a chip, they're going down exponentially, following Moore's law pattern. So photons travel faster than electrons. Photons also do not heat. Electrons get hot. So right now, one of the problems, it's both lithography, but then it's also heat dissipation. So how do you get the heat out of your electronic circuit? Which is why a lot of supercomputers require liquid nitrogen cooling, because the processors get hot and then you have to cool them, otherwise your processors melt. Kind of like your cell phones, at least old cell phones a couple years ago, used to get really hot in your pocket. So now, we're looking at doing basically optoelectronic circuitry. So you use optics, which does not heat, to carry a lot of the information, and then you do some of the processing with electronics. So you basically get the best of both worlds. So in order to create an optical circuit, you basically need a lot of the same building blocks. So you need waveguides, which are the optical equivalent of a wire. Couplers, which is basically how you can control where your photons go. So it's basically waveguides that merge together and then split and divide light in different paths. And then resonators, which basically form either memory storage circuits or they form the foundation of lasers. So it's 3 building blocks, kind of a toolkit. So in our group, we're making all 3, coming up with new ways actually making all 3. So the first is the waveguide. So these are made out of glass. So glass is a really great material in the sense that it's transparent from about 300 nanometers all the way to about 2 microns. So a micron, for those who do not know their SI units off the back of their hand, is 10 to the minus 6, and a nanometer is 10 to the minus 9. So that's a very, very wide range that you can shine light through this waveguide. So you can make them in lots of different geometries, lots of different patterns, squiggly shapes, straight shapes, things along those lines. We can also take them and change the pattern and then create couplers, so then we can controllably change the path that the light is taking on the silicon wafer. And then we can also make resonators, which form the foundation of lasers for the integrated photonic circuits. So how we change if we go to the left or the right or both, is there's actually material in this middle region which changes the refractive index. So it's lithium niobate, which is a nanoparticle, and it responds to electric fields. So we can change in real time as we apply an electric field, so we can modulate how that refractive index, or the value of that refractive index, and then we can change if it goes left, right, if it's what's called a 50-50 coupler, which means it splits exactly 50-50 like is shown here, or if it's 90% one way, 10% the other way, or however we want it to go. So we use a process called photolithography. So how photolithography works is it's kind of like developing film. So you start off with your wafer, and then you spin on a photosensitive polymer film, kind of like if you were, when I say film, I'm talking conventional, like 1980s film. So you have a photosensitive film, and then you put what's called a mask on top of it, so it's just a pattern with some aspects of it are clear and some are black. And then you expose it to UV light, and then develop it like you would develop film. So then you end up with some parts of your silicon wafer having parts of the resist still there, and some parts where there isn't any resist anymore. And then you transfer the pattern using a buffered oxide etchant, so basically HF, because HF etches silica and doesn't etch silicon. And then you remove all of your resist, so that now you have your pattern etched into your silica, but not etched to your silicon. And then you use an etchant that will etch into your wafer. And then you end up with that. It's actually really, it sounds really hard. This is actually probably the easiest device, because it's one lithography step and 2 etching steps, which is really easy. So the resonators, you put in a single pulse of light, and the light is confined in a circular orbit, so it basically orbits around. Depending on how good of a resonator you have, it can actually orbit around as many as 100,000, 200,000 times. So if you have a really good one. If you have a bad one, it doesn't stay in at all. So because it is able to be confined in there so long, you end up with these very high buildup factors. And so then because you have such a high intensity orbiting optical field, you're actually able to turn this, what would otherwise be just a normal glass device, into a laser. So you're actually able to take one input wavelength and get it to generate light of another wavelength. So you're able to use these to basically create an optical upconversion effect and get lasing out of what would otherwise be a passive silica device. So we use them to make lasers. You can also use them because they do store light for long periods of time, so on the order of a couple microseconds, which sounds really short. It's really long. Okay, so it sounds really short because it's like a microsecond, right? But photons travel really fast. So a microsecond's really long for a photon because photons travel really fast. So you're able to use them as optical memory. So DARPA is interested in using these for optical memory. And keep in mind, DARPA is like the really cool government agency that thinks like, 20 years in advance. So if DARPA is interested in them, then we all should be interested in them. Okay. So they're thinking about them for optical memory. Other people are thinking about them for lasers. So there are a couple different ways. So here I just drew a free space light beam going in. We use integrated waveguides. So we actually fabricate this type of waveguide and this type of device simultaneously. So then we can just couple light from this waveguide into this device. You can use free space. So you just shine it and it just couples in. You can also use something called a tapered optical fiber waveguide, which is where you take a piece of optical fiber, you heat it in the middle with a hydrogen torch, you pull on both ends, and then it melts, so it's kind of like pulling glass. And as long as you pull it until it's smaller, so the diameter is smaller than the wavelength of light you're using, so if you're testing at 1550 nanometers or 1.5 microns, then the diameter has to be a micron or smaller. Then you can also use that to couple light in. So it depends what you want to do. Which approach is easiest, right? So the uber quick and dirty approach is just to shine light. The next easiest is to tapered optical fiber. And the most robust, but perhaps hardest, is to actually integrate the waveguide onto the wafer. So most of the students in my group go with the tapered optical fiber approach.
Does that mean they are pulling one by one
Yep.
to produce single resonators?
To produce the waveguides.
Okay.
Yeah, all the resonators are fabricated kind of like this, so we end up with somewhere 10 to 20 on a single chip. A chip being a couple millimeters by a couple millimeters. Otherwise, you can't hold them.
But eventually you want a million of them on a chip, or tens, whatever.
Yeah, probably more like 20 or 30. You don't really need that many.
Oh.
Because it depends what you want to do with them, right? But you're not going to need that many. So on the chem biodetection side of the group, there are kind of 3 overarching themes. We're working on developing some biomimetic coatings, which are things like lipid bilayers. That's the fancy name, biomimetic. studying substrate enzyme binding kinetics and looking at polymer thin film behavior. So I'm going to go through each of these briefly. So a lipid bilayer is called biomimetic because it is a good model for a cell membrane. So cell membranes are lipid bilayers with other things in them. So this is a cartoon of a cell membrane, and the lipid bilayer structure is that head, tail, tail, head portion of this whole complex structure. Of course, there are other things in a true cell membrane that are not present in a lipid bilayer, like the glycoproteins. Usually there will be, are they in here? No, they aren't. Usually there are also like sodium transport channels, things along those lines. But the actual lipid bilayer is a pretty good model for studying how proteins can move throughout the cell membrane. So people who study how cells behave and are interested in membrane dynamics use lipid bilayers. They're also very, very easy to make. So there are a couple different routes you can use to make them. You can use a solid supported lipid bilayer, which means that this structure is actually supported on one side by a surface, like a glass surface or a silicon wafer or something along those lines. Or you have a suspended lipid bilayer, which means that it's anchored on either side, but then it's free floating in the middle. So there are positives and negatives to both approaches, like always. If it's anchored on both sides, then at the anchoring edges, you can end up with edge effects, which will distort and the behavior won't be the same as you would find in a true lipid bilayer. If you have the solid supported bilayer, then there is a chance that at the solid lipid interface, you could have edge effects. So there are positives and negatives. What we're working on doing is creating self-assembled, oh, I forgot to mention, forming the lipid bilayers is a self-assembly process. So unlike making our devices where we actually have to pattern them, the lipid bilayer structures naturally form. So you put a bunch of lipids in the correct concentration in a solution and sonicate it and then let it sit for 36 hours to 48 hours and you get a lipid bilayer. It's awesome. Like there's no, it requires patience, that's it. So this is how we do it. So we actually have microsphere resonators in this case. So instead of the little toroid donut shapes, they're spheres. They work the exact same way. We use spheres because for this particular experiment, I actually had an undergrad do this, and you can make spheres without going in the clean room, whereas to make toroids, you have to go in the clean room, and therefore we did spheres. Yeah. So we have spheres, and we made the self-assembled lipid bilayers on them, and this is an optical picture, not an SEM. So it's like the normal standard wide field picture, and this is a fluorescence picture. So the reason why this picture is significant is because we actually put a fluorescent dye in the bilayer, and so then we showed that we basically had this very nice uniform bilayer all over the surface of our resonator. So this is kind of nice, and it's a nice cute little optical trick. But what's really cool is we could actually monitor, using the optical device properties itself, basically the bleaching time of the dye in the bilayer. So we can actually monitor the photodynamics of the dye within the bilayer environment. So we're now going and we're putting in transmembrane proteins, and we're looking at how different proteins go across the bilayer. So we're actually studying membrane dynamics. So actual fundamental bio problems that people haven't been able to study. Because with fluorescence microscopy, you get very good resolution in the XY plane or within the bilayer, but not in the Z plane. And we're able to detect the Z plane. So there, in studying any protein interaction, so this could be antibody-antigen, substrate ligand, anything like that. So wherever you have a protein and a receptor molecule, so two things that are meant to stick together, you end up with a curve that looks something like this. So as you increase as you monitor how the two molecules bind together, so this is time and this is whatever your detection signal is, you're going to end up seeing the two molecules bind until they hit some sort of equilibrium saturation point, and then they will begin to unbind. So this binding region describes how strongly they're attracted to each other. The equilibrium region is basically determined by the concentration of molecules in the solution. And then the unbinding region is described by, or is determined by, the strength of the interaction with the molecules. So from this simple curve, from the binding and the unbinding, you can determine a lot about the strength of the protein-protein interaction, the density of the molecules on the surface, the pH of the environment. So the reason why people are interested in determining the binding and unbinding profile is because in drugs you want to have the strongest affinity, at least for most drugs, you want to have the strongest affinity between your target drug and whatever the target molecule is on the surface of your cell, especially in a lot of the new designer drugs, which, let's see, what's a good example? There is a monoclonal antibody drug for colon cancer, which is basically designed just to target VEGF. And so it only binds to VEGF on, which vascular endothelial growth factor. It's a protein. And it binds to VEGF with a very high affinity. So it binds and sticks very, very efficiently. So in order to determine if their drug does this, has this binding behavior, they need to be able to detect it and record it. So they would do a test like this and see exactly what that binding affinity was and what the off rate was. And obviously, you don't get a drug right the first time. You make a lot of mistakes along the way. So one of the things that we're working on is developing sensors which can perform these types of experiments. Because the sensors that they use currently are effective, but they have problems at low concentrations. And when you're doing drug trials, you don't necessarily want to have to make things at micromolar concentrations. You'd like to be able to make things at femtomolar concentrations, so lower. Femtos 10 to the negative 15. So then the last thing is polymer behavior. So ultra-thin film polymer behavior. So thin film stops around 500 nanometer, and ultra-thin film is around 300 nanometers or below. So very, very, very thin films of polymers. So what we do is we take one of our toroid devices and then we coat them with a very thin layer of polymers. And we do this a couple different ways. We either do it the brute force way, which is spin coating them, which spin coating is kind of like spin art, if any of you did spin art as a child. So spin art is when you would take a piece of paper and put it on something that looked kind of like a record player, and then you would turn on the record player-esque item, and it would start spinning fast, and then you would like squeeze paint on it. So then, because it is spinning, your piece of paper is spinning very, very fast, then your paint would smear out in a splatter paint-esque fashion. And you would obviously not just put blue paint on your spin art, you would put blue and red and yellow and green and many different colors, and then you would end up at the end of the day, when you hit stop, having smeared paint in a very 1970s, 1980s like fashion. That was really cool. Apparently now, because radio players aren't in fashion anymore, neither is spin art. Anyway, so spin coating, which is based on the same principle, involves putting your wafer on a piece of equipment which spins in a similar fashion to a record player, except it spins at about 6,000 RPM, so much faster. As a result, you get a much thinner film, so 100 nanometers or so, or less. We can get down to 20. And very uniform, because it's spinning so fast. You get very uniform films. So usually there's about a 10 angstrom or 1 nanometer variation across the film. So it's very even. So that's spin coating, right? As you can see, it's very fancy. It's actually the way that Intel uses to put down their polymers. So it's very accurate. However, it's also kind of the brute force way, right? A 3-year-old can do spin art. Given the appropriate training, in other words, the spin coater involves a pedal on the ground to start it and a different pedal to stop it. Okay, so given the appropriate pedal training, a 3-year-old could probably use a spin coater as well. The other way we do is called grafting. So we actually start off with a monomer unit of the polymer, so just a single subunit. So like this, and then we actually grow it out from the surface of our device, kind of like a chia pet. So you grow out from the surface of your device kind of like a chia pet. And you end up, just like with the chia pet, having very, very, very dense arrays of polymers. And the polymer chains should be somewhat parallel to each other and not really get tangled up or interact or anything like that. And it forms this very, very nice brush structure. And doing it that way is actually called a polymer brush. I did not coin that term. So then we can actually compare the results we get via the spin coating brute force approach with the chia pet approach. So this is just some sample data. So basically, as we change the temperature, we can actually monitor how the refractive index changes, so the effect of temperature on refractive index. Depending on if we have a spun coat film or polymer brush film, we get very, very different behavior. So if we have a spun coat film, we actually see a positive change in refractive index. If we have the polymer brush film, we see a negative change in refractive index, which is just uber cool. And then depending on the film thickness and depending on what wavelength we measure, we get a different glass transition temperature. So the glass transition temperature is a parameter of a material, of a polymer material, that describes when it goes from being solid to being viscous, so somewhat liquidy. So the glass transition temperature is one of those critical polymer material properties that you want to characterize. And there's clearly a film thickness dependence, which is known, but what this data shows is that there's also a dependence on how you're characterizing it. So it means that depending on how you decide to measure it, you're going to get a different value, which is very important. That's like saying you could be going 60 miles per hour, but it depends on if you decide to make the measurement from 10 feet away versus 20 feet away. You're going to get a different measurement, but you're still going 60 miles per hour. So that's what this data is showing, is that clearly this polymer only has one glass transition temperature. It's the same polymer, it was the same device. We measured it on the same day, we just used different lasers. But we got very different glass transition temperatures. So that's important, right? Because you need to know what the TG of your polymer is. So on some of the, I didn't talk very much about it because we only have preliminary data, but we're working in a field called epigenetics, which is basically studying so DNA normally does not have methyl groups on it. So the base pairs don't have extra methyl groups. But it's thought that there's a link between the number of methyl groups you have attached onto your base pairs and both your risk of getting cancer and if you actually get cancer. And both the number of methyl groups you have and their proximity to each other. So it's not just an overall, if you have, let's say, out of a short segment of your DNA, let's say like 20 base pairs, if 5 of them are methylated, right? Maybe it's not that you have 5 methylated ones, maybe it's that all 5 are close to each other. And maybe it's not that all 5 are close to each other, maybe it's that all your T's are methylated. Maybe that's significant. Because research in like 2007 showed that there was a link between just the existence of methylated DNA and an increase in prostate cancer, colon cancer, breast cancer, basically almost every type of cancer. But they couldn't tease out all of these different effects, right? Is it a proximity effect? Is it a type of base pair effect? Is there a given threshold? But they could tell that it was significant. And the reason why methylation is important is because if you have a methylated base pair, then that is passed on to your children, which could explain some of the genetic links. Because if I have methylated base pairs, then I'm going to pass it on to my child. But then also, as I get older, my DNA continues to get methylated. So then it's basically never going to get better. Not to make all of you very depressed. But you basically have all the problems that your parents had, plus any others that you decide to pick up along the way. So it also could explain why the prevalence of cancer is increasing over time, right? Because it's clearly not going to go down because our cancer or our DNA is continuing to get methylated and it's not getting demethylated. It's not going the other way. So there's kind of two. So Keck has a gigantic epigenome center, which is, I'm going to get the number wrong. It's in the $5 to $20 million range, funded by NIH. So I'm working with some of the researchers there. They're working more on the trying to understand a lot of the links in the monkey, rabbit, mouse models with some human samples using conventional detection methods, and I'm working on coming up with better and faster ways to understand the links. So their ways, they get a sample and it will take a month to 2 months to analyze a single sample, and I'm trying to come up with a way that will take an hour. So then it will just be faster, because it should be faster, basically. So what we're working on, obviously once we understand exactly what causes what, then coming up with ways to demethylate your DNA. Right after you can clean it up. So some drug companies are working on coming up with ways basically to go through and strip your DNA of all the methyl groups. But before they do that, they need to understand what the consequences of that would be. All of this is very unknown. It's a completely new field.